times, I thought I'd give you a title that maybe you can relate to, because the last year or so has been particularly stressful for many, many people. And so I'm going to talk to you about what happens when we are all stressed and how that relates to DNA damage, aging, and antioxidants, and the work that my group here at Clemson does. Now, I said, or in the introduction, said that there were going to be a bunch of questions in my talk, and there are three major questions that I want to try to answer for you tonight. The first is, how is DNA damaged when people are under stress? The second is, how is this DNA damage related to diseases and aging? And the last one is there anything we can do to prevent this damage? And so I have a talk that's maybe about 30 minutes, but if you have a question and we get to a slide like this, feel free to ask. And I will do my best to answer. So the first question is, how is DNA damaged when people are under stress? And this is a really well-known process that happens. It leads to some noticeable consequences. And so I have provided some visual representation of what happens when people are under stress. And you may recognize these people. And this is what happens when you are under stress. These are the same people, 8 years apart, right? And you can notice what people might think of aging, premature aging. And I don't have to tell you that being president of the United States is a particularly difficult job, and it results in a lot of stress. And you can actually physically see this stress as it, occurs. And I don't mean just a job stress. When I say stress, what I mean is anything that is unfavorable to an organism, right? So if you're up really late, you don't get a lot of sleep, you have insomnia, something like this, you are under stress. If you have chronic disease, your body is under stress. If you have some disease treatments like chemotherapy, your body's under stress. If you're just worried all the time, that's another stress. So all of these things are under this large umbrella of what I consider stress and what people study in the field of oxidative stress. And it's related to a lot of different diseases. So at the center is this stress, and depending on where this stress happens in the body, you can have different effects. So in the brain, for instance, you can have strokes that are a result of oxidative stress, or Parkinson's or Alzheimer's diseases, aging over here, cancer, diabetes. A lot of these diseases of aging, that people associate with aging, are a result of stress on the system. And so you can pretty much pick your favorite organ and see what happens when it's stressed. So if you look at the system from a cellular level, so this is my diagram of a cell. So you have this right here is the cell membrane. Over here, you remember your biology. This over here is the nucleus with some DNA in it. There's a mitochondrion with some DNA in it. And all of these, all of the cells have natural antioxidants, molecules that try to keep things balanced in the cell. And what they're balancing is called redox potential. And so these all have acronyms, NADPH and glutathione, that's what this GSH1 is, and cysteine over here. But the idea is that these are a redundant system of trying to keep your cells in a very particular environment so that the processes that need to happen for life can occur. When that gets out of balance, if you have a depletion of these natural antioxidants, or if you have metal ions like iron or copper that increase in their cellular levels, then you get oxidative damage, which can occur to DNA or proteins or these membranes or anything. I'm going to focus on DNA today, but that cellular damage can actually lead to those diseases I showed on the earlier slide: cancer, cardiovascular disease, aging, neurodegenerative diseases. All those kinds of things are traced back to oxidative DNA damage. And so there's a lot of interest in dietary antioxidants, whether or not, if you eat a lot of dietary antioxidants, maybe they can make up for depletion of these natural cellular antioxidants. And one of the things that I've been looking at is, do these dietary antioxidants bind these metal ions like iron and copper? I got my little laser pointer going on. Do they bind those metal ions to prevent this kind of stress? And so what we know right now is that iron, here, this is iron, and hydrogen peroxide over here, if you combine those two and in the reduced form of iron with hydrogen peroxide, you get hydroxyl radical over here. This OH dot is hydroxyl radical. And that is, if you've ever heard of damaging radical species or free radicals that cause problems, this is what they're talking about. This is what people are talking about. And hydroxyl radical will damage just about anything it comes in contact with in the cell. And so you have this problem where you have the iron and you have the hydrogen peroxide, and you have radicals, damaging radicals being formed, but it's worse than that. The cellular reductants here, I've shown one of them, they can actually take, the cellular antioxidants can actually take iron 3 and reduce it back to the iron 2 that creates more radical species. So this becomes kind of a circular or catalytic process where you keep generating hydroxyl radical, you keep generating these damaging species. And so this iron-mediated DNA damage is the primary cause for cell death. This whole sequence of events, for everything from E. coli bacteria to humans. So you might think, well, why don't you just get rid of the iron? Unfortunately, we need iron. We need iron to do a lot of things. You know, iron is in hemoglobin, and it carries the oxygen around in our body. So if we remove the iron from our body, we would die a lot faster than this oxidative stress kills us. And you might say, well, how come we have so much hydrogen peroxide around? If you lowered that, wouldn't you not make so much hydroxyl radical? And that's true, except that hydrogen peroxide is a byproduct of respiration. You know, if you're breathing, you're making hydrogen peroxide. And so unfortunately, not breathing will also kill us much faster than this oxidative damage process. So we're kind of caught, you know, in this cycle where the two things that will help us live, this iron and this hydrogen peroxide, or, you know, respiration, also kind of slowly kill us. So there it is. But it turns out the iron binds to DNA really well. And there is a piece of the DNA double helix here. You can see the sides of the double helix here. And in between here, these red things are base pairs. And over on the other side, over here is just a blow up of that picture so that you can see the base pairs better. And this little orange thing that looks like a jack, that's actually iron. And this is a structure of DNA that was solved with iron binding to the DNA, to this, it's a guanine residue right here in the DNA. So that's the G base of DNA. And so you can imagine if hydrogen peroxide would come around to this iron, it would generate a radical species, a hydroxyl radical, right next to the DNA. And that hydroxyl radical would damage the DNA and break it apart. And that's exactly what happens. The iron localizes on the DNA, and that's where you get the radical damage. So this is a problem. Fortunately, your cells have a way of repairing DNA damage. You know, they can repair or prevent some of this damage from occurring. But the problem comes when you get more damage than your cellular processes can handle. Then bad things start happening. And here's another way to look at the same process. This is actually what's called supercoiled DNA. It's wound up tightly. You can think of it like a rubber band if you're just winding up a rubber band and holding both sides. And if you add iron and hydrogen peroxide, that causes a break in the DNA backbone in the double helix, and you can't hold that supercoil anymore. It unwinds in sort of a floppy circular piece of DNA here. And if you add even more iron and hydrogen peroxide, you can actually break this DNA up into linear bits like this. So this is sort of a visual representation of the DNA damage that's going on. And people have studied primarily iron and copper for this process of DNA damage with hydrogen peroxide, but a lot of other metals are capable of generating radical species that damage DNA. And I've listed a few here: manganese, nickel, cobalt, chromium. And so this is an active area of research, how these metal ions cause damage and what might prevent it. And as I mentioned, all of these metals, iron and copper and all the rest, are implicated in a lot of these diseases of aging because of their ability to damage DNA. So this is an example of DNA gel electrophoresis. And this is a method that we use to look at how DNA is damaged and how antioxidants can prevent this damage. So I thought I'd show you a gel here. The gel itself is actually like Jell-O. It's a purified form of seaweed. You could probably eat it, although it wouldn't taste very good. But the DNA can kind of wiggle through the holes in the gel, and you can separate out the different forms of the DNA, damaged and undamaged DNA. This bottom band here is the plasmid DNA that's not damaged. So anytime you see it down here, you know that it is intact. And when you add hydrogen peroxide by itself, still not damaged. There's a little bit of damage, but that happens when you isolate DNA. When you add iron and hydrogen peroxide, you start to see a little more damage. And as you increase the concentration of iron here, you increase the amount of damage. Like this is all with hydrogen peroxide. So as you increase iron, you increase the amount of damage. So great. She just told us in 3 different ways: more iron damages DNA, more. Okay, but so that gets to the second question here, which is, how is this related to those diseases that I talked about and to aging? And if anybody has any questions so far, feel free to ask. And if you don't, that's all right, we'll go on. So if this DNA damage, as I promised, is related to aging, well, I've got to explain a little bit more about DNA structure. You know, here's the double helix of DNA, and it winds itself around into these coils. These purple balls are just proteins that it winds around to try to keep things from getting tangled. And these coils then wind around into supercoils that then kind of conglomerate into these chromosomes, right? And when you have all this DNA wound up quite a lot, it really does make a big difference in how it packs into the nucleus. You can see the nucleus diameter here is about 6 microns. And you have in humans, we have about 3 billion base pairs of DNA. So if you extended that all out, it would be about 1 million microns. So you can see just how packed the DNA has to be in that nucleus to fit. It's about 2 meters of DNA in every cell. And once it's packed into the chromosomes, it's also protected a little bit from all of the things that could damage it. But I do want to point out that these chromosomes have ends. Each one of these, 4 sites has an end of the DNA. And so then it begs the question, well, what happens at the end of the DNA? And the answer is there are these structures called telomeres. And these are a fairly recent discovery. This was the Nobel Prize in 2009 that these 3 researchers shared for the discovery of telomeres and how they protect the ends of the DNA from degradation and damage. So this micrograph here is actual chromosomes, and there has been added to the chromosomes a fluorescent marker. You can see that binding to the telomeres and lighting them up. So everywhere there's an end of the DNA, you see one of these little white dots. And over here in my picture here, you see the telomeres, they're purple, the chromosome is orange. And so you see the telomeres at the end of the chromosome. And these telomeres are thousands of repeats of the human telomeric sequence, TTAGGG. So those are just the base sequence for these telomeres. It repeats thousands of times. And when the cell divides, you get one cell, it divides into 2, the 2 divide into 4, the 4 divide into 8. Every time the cell divides, those telomeres get a little bit shorter and a little bit shorter and a little bit shorter, because it's hard to copy the DNA all the way out to the end. And so when the telomeres get too short, cells undergo a process called apoptosis, which is programmed cell death. So this is a way, the telomeres also are a way for the cell to sort of keep track of how many times it's divided and when it is time to die. And hopefully, when it is time to die, the cell dies. If you don't have cell death when it's supposed to happen, you get things like cancer occurring, where cells just overgrow. They don't have a way to shut off their growth. Okay, well, that's nice to know, I guess, but how is it related to aging? And to do that, I have to show you a little bit more of the DNA results that people got to look at where DNA was damaged when you have hydrogen peroxide with iron. And at low hydrogen peroxide concentrations, you see damage at places like ATGA sequences and GTGG sequences. At high hydrogen peroxide concentrations, on the other hand, you have damage at AGGG sites. So high hydrogen peroxide, high stress conditions, you see AGGG damage. And so you get damage at these AGGG sites. Unfortunately, AGGG sites are found in the telomeric sequence. So you have thousands of repeats of this sequence where iron can localize and damage the DNA. So iron localizes and damages the telomeres. And this is actually a good thing. It's a protective thing, because you can see the iron here. This is DNA double helix again. These are guanine bases in the GGG sequence, and you can see the iron bound to the guanine bases here. They know that iron localizes on these because iron localizes on guanine. Since you have many thousands of these AGGG sites where iron localizes on the telomeres, you get a lot of DNA damage at the telomeres. And so the telomeres actually act sacrificially to prevent damage at the coding parts of DNA, the DNA that's required for cellular functions. Telomeres are just protective ends. So if they get damaged a little more, that's okay, as long as the DNA that's supposed to do things can still function. But the telomeres get damaged and they get even shorter. And so the cells die. And if you have more stress, the telomeres get even shorter and even more cells die. And this is the oxidative theory of aging, how aging happens. When the cells, when more cells start to die than are reproduced, you know, that are born essentially, then you have aging. And this is the prevailing theory of how aging happens. It is iron-mediated DNA damage. So it's a cheery story, isn't it? Aging is a cheery story. But the question then is there anything we can do to prevent the damage? Because I mentioned the cellular antioxidants that are supposed to help keep this all in check, right? They're supposed to stop the damage. But what about those dietary antioxidants? Can we prevent the damage with dietary antioxidants? Well, that's where my research comes in. And we've been looking at this question, how do antioxidants prevent DNA damage by metals, for a number of years now. You see that we look at DNA damage prevention. We look in cells, this is E. coli here, how antioxidants can rescue cells from oxidative stress and metals. And we actually also do some synthetic work looking at how antioxidants bind metal ions like iron here. And this structure is an iron structure with some antioxidants attached. Now, one of the antioxidants that people talk a lot about are phenolic antioxidants. These are the ones in fruits and vegetables and green tea, and as you can see, dark chocolate and all of this. This is one of the more common kinds of antioxidants. There are about over 10,000 phenolic type antioxidants known with separate structures, and they share characteristics. These catechol groups with 2 OH groups here, if you're interested in chemical structures, or they could have 3 OH groups on them. And the oxygens here bind the iron. So we know that these phenolic compounds bind iron really well. And so what I was wondering is that the reason why they're good antioxidants? And when I started over 10 years ago, nobody knew the answer to that question. They didn't know if iron binding was the cause of these antioxidant activity. But since I haven't had any questions for you, I'm going to ask you a question. Does anybody know what the primary source of antioxidants, phenolic antioxidants in the American diet is? Nobody willing to, did I hear somebody? What? Coffee.
Coffee.
Yeah.
Yes, it's coffee. So we are down here. Average source of phenolic antioxidants in the American diet is coffee. We should be up here somewhere, maybe, if we're a little more healthy about what we eat. Although I do want to point out that although dark chocolate is up here at the top, there are many nutritional reasons besides the antioxidants why eating a dark diet where dark chocolate is a primary constituent may not be a good idea, right? You can go overboard with this sort of thing. And so we looked at a variety of phenolic antioxidants, and here are the structures. And for those of you who are not familiar with the structures, that's okay. I've divided them into this class, which are the small ones, and this class, which are the big ones. And that's all you really have to know. So the small ones, these are food additives, and you know, to protect food from oxidation, antioxidants in food. And in fact, if you go and look at a box of crackers or cereal or something, you may see this N-propyl gallic acid or gallate as an ingredient to preserve freshness. These other ones, the larger, more complicated ones, these are the phenolic antioxidants in green tea. And I'm going to talk about this one here, EGCG, a whole lot from here on out. We're going to focus on that one. But just remember that it's from green tea. You don't have to remember the structuring exactly. There will be no quiz. So we're going to test this green tea antioxidant, EGCG, for its ability to prevent iron-mediated DNA damage. So here's another gel electrophoresis figure. And you can see the DNA here, down here in the lower band here is undamaged. And when you add the iron and hydrogen peroxide, you see that it is now the upper band, and this is damaged DNA. And then as you increase the concentration of EGCG, this green tea antioxidant, you see that this upper band, the damaged band, gets less intense. So it's preventing the damage. And until at the very last lanes over here, you see no damaged DNA. All of the DNA is undamaged. So this green tea antioxidant can prevent iron-mediated DNA damage at low concentrations. And we know also from other studies that iron binding is required for this damage prevention. Because if you use iron that's fully bound with something else to prevent the EGCG from binding iron, then you don't see this kind of effect. You don't see a protective effect with the DNA damage. So once we have the gel electrophoresis, we graph the percent inhibition of DNA damage. Right, you start at 0, you end up at 100% inhibition of damage. It prevents, you don't get over 100% prevention of damage, versus the concentration here, in this case of that green tea antioxidant. And so we use these graphs to determine what's called an IC50 value, which is the inhibitory, the concentration required to inhibit 50% of the DNA damage. Right, so at this concentration here, it prevents 50% of the DNA damage, and that concentration is about 1 micromolar. Now, the concentration isn't that important. What's more important to understand is that the more effective an antioxidant is, the lower this inhibitory concentration is. So lower values are better. They prevent more DNA damage. The other thing to know is with green tea antioxidants, if I were to drink a cup of green tea, does anybody want to venture a guess in 1 hour what my blood concentration of green tea phenolic compounds, you know, antioxidants would be? Nobody's brave enough? About 1 micromolar. So that's with 1 cup of green tea after an hour. So these are very relevant concentrations that we're seeing this damage prevention. And so I have my nice little bar graph so you can see the IC50 values here. Remember, lower is better. So EGCG is the best one that we tested of all of the antioxidants that I showed. And you notice there is a fairly wide range of activity for all of these polyphenol antioxidants, even among, you know, the polyphenols in green tea. You'll also notice that a lot of them prevent damage with IC50 values of about 10 micromolar or less. And that's about the concentration, the maximum concentration of polyphenolic compounds in your blood. If you eat a diet rich in fruits and vegetables and foods with polyphenols, then you get a concentration in your blood about 10 micromolar. So a lot of these will prevent DNA damage at those concentrations. So that's good. Woohoo! We have you know, a biological antioxidant, dietary antioxidant that can prevent biological damage, at least at the right concentrations. So we wanted to see if we could find a property of these polyphenol compounds, because I said there were 10,000 of them. How do you pick the best ones? Well, we thought, what properties relate to their iron binding? Because we think that that's what's controlling it. And we found this pKa property, which is basically a measure of how well the polyphenol can bind to iron. So the more acidic phenol compounds bind iron better, and you can see this nice correlation that more acidic polyphenols have lower IC50 values. They're better antioxidants. And the great thing is you can calculate this too. So you can take all those structures of those 10,000 polyphenol compounds and calculate which ones should have lower pKa's and which ones should bind iron. So this is the first predictive model for polyphenol antioxidant activity, and it's based on metal binding. So we went, woohoo, we're on the right track. But there are a lot of other antioxidants. So I want to tell you just the conclusions for this, and then one other example of antioxidants. So, you know, iron binding controls polyphenol DNA damage, and we now have predictive models. But, and this is very important, although polyphenols prevent iron-mediated DNA damage really well, unfortunately, when you do the same studies with copper, these polyphenol compounds can generate radical species and enhance DNA damage. Boo! So things are really complicated. And we have to test all of these antioxidants with each of the metals. It's not a you know, general mechanism that we can say. But I just wanted to show you here what happens when you have a polyphenol like EGCG with iron and in oxygen just in air, because it's actually kind of pretty. So it starts off colorless. And then after a while, as the iron binds and oxidizes, you see it turns a very nice, pretty purple color. So these compounds really do bind iron quite well. Okay, I promised one more example of antioxidant compounds. So I'm going to switch to sulfur and selenium antioxidants, totally different kind. You can see some structures here. They look nothing like the polyphenols. And this is glutathione. This is one of the natural antioxidants in cells. And these compounds are found in high concentrations in cells, very high concentrations. And you get these sulfur and selenium antioxidants from a variety of dietary sources: garlic, onions, and other members of the allium genus. Brazil nuts also have a lot of selenium because they're grown in selenium-rich soil. And there have been a variety of clinical trials to look at selenium supplementation and how it would prevent disease. So I'm just going to show you two examples to illustrate the problem with dietary supplement trials. This nutritional prevention of cancer trial, people were given 200 micrograms a day in the form of selenium-enriched yeast, which is mostly selenium methionine. And they found that in this trial, people had 37% fewer incidences of cancer, particularly prostate cancer. So people were like, whoa, 37% fewer incidences of cancer, that's amazing. Now this was a small trial. It only had about 1,000 people in it. So they said, we got to do a bigger trial and make sure that this is real. And so in 2008, a trial called the SELECT trial, it was the largest nutritional supplementation trial ever done. $200 million, 10,000 people. And it was halted, it failed. It was halted in year 7 of 12 because when they gave people 200 micrograms per day selenomethionine, it did not show effects. And so that was a big bust. And there are a number of reasons that people have put forward why they had two different results, very different results from these similar trials. The first was that the first trial used selenium-enriched yeast, which is only 80% selenomethionine. So how do you know that selenomethionine is actually doing, having the effect and there's not something else in there? The other problem was this nutritional prevention of cancer trial was actually conducted in North Carolina, where there is selenium poor soil. And so you'd expect people in North Carolina to have less selenium in their blood than average. Whereas the SELECT trial was conducted in Arizona. And in the West, there is a problem actually with high selenium levels in the soil. They can be so high they can kill grazing animals in some areas. So you have two different areas, and it's possible that the people in the SELECT trial had much more selenium in their bodies than in nutritional prevention and cancer trial. So that's why selenium supplementation helped in one trial, but did not in the other. So we tested all these sulfur and seleno amino acids for their ability to prevent DNA damage with copper and with iron. And you can see here that with iron, you don't see a whole lot of activity. Not those nice low IC50 values that we've seen before. With copper, however, the story is different. All of these compounds prevent a lot more copper-mediated DNA damage than iron-mediated DNA damage. But you'll notice selenomethionine has the highest IC50 value of all of them. And that's the one that that's the compound they selected for that trial, that clinical trial that went bust. So maybe they were looking in the wrong place. Maybe. We don't know yet. But it is pretty clear that these sulfur and selenium containing amino acids prevent mostly copper-mediated DNA damage. They do that very well, but not the iron-mediated DNA damage. So they're opposite compared to the polyphenol antioxidants. They have opposite activity. And again, we wanted to see whether this activity was due to copper binding. Unfortunately, amino acid binding to copper was not really known for these sulfur and seleno amino acids. So we did a series of studies to show that the copper actually is bound to the selenium or sulfur of the amino acid, as well as the amine part of the amino acid. And once we had this structure, we could then calculate the energies of these compounds and how easily they could be antioxidants. So this is the copper with the amino acid in the water here, the energy of the complex, and you'll see that this correlates quite well with the IC50 values. So again, we understand how these sulfur and selenium antioxidants can be preventing copper-mediated DNA damage. We've even developed a predictive model for this activity. So this is the first predictive model for this class of antioxidants. So this leaves us with only one question, which is, why do these amino acids, these sulfur and seleno amino acids, prevent copper but not iron-mediated DNA damage? And we just recently completed a series of studies. This is penicillamine. This is an amino acid. There's the sulfur there. And it binds both copper and iron. And but it is a drug. It's a sulfur-containing amino acid drug that's used to bind copper to treat people with copper overload. They have too much copper in their bodies. It's called Wilson's disease. And so they're treated with penicillamine that binds the copper. And once the penicillamine binds copper, you can excrete the extra copper through urine. But what we found is in our model that even if you have 10 times more iron than copper, you still see all of the penicillamine essentially bound to the copper and not the iron. So penicillamine binds copper. These sulfur and selenium amino acids bind copper really strongly. They don't bind iron very strongly at all. And that's why we think that the amino acids don't prevent iron-mediated DNA damage, because they don't bind to iron strongly enough to show those effects. So where does that leave us? Well, metal ions can cause a lot of problems. They generate radical species that damage DNA, they kill cells, they lead to a lot of diseases and aging. And trying to figure out how you can combat that is complicated, right? Antioxidants are metal specific. You have different classes of antioxidants that prevent DNA damage by different metal ions. So you have, of course, the sulfur and selenium amino acids that prevent copper-mediated DNA damage, the polyphenol compounds that prevent iron-mediated DNA damage, but with copper, they're pro-oxidants, they cause the damage. So you really have to be careful, that you understand what's going on. We have been able to develop predictive models for antioxidant activity to help figure out which antioxidants might be best for some of these clinical trials that people want to run with dietary antioxidants. But it's a really complex thing. You're not going to have a magic bullet, the antioxidants that's going to cure aging. Unfortunately, we're a really long way from that because it's a really complicated system and it's not a one-size-fits-all solution. So that's my seminar. Of course, I have to acknowledge all of the people who helped with this work, my former and current graduate students, collaborators over the years, and of course the funding agencies for supporting this work. And I'm so glad to be able to show it to you. I'm sorry I couldn't be there in person. I really, sitting in a chair and giving a seminar is not my favorite. I really like to be walking around the room, waving my hands around. So, but I'd also like to answer any questions you might have.
Yes, I do have hopefully a simple question. First, thank you for an amazing presentation and how you dissected these components. My biology was not my strongest, but you're an amazing person in communicating and simplifying it. So can you contrast black tea versus the green tea in terms of the antioxidants capability? Mm-hmm. I saw that the black tea had much more antioxidants than the green tea. But I was puzzled by why you chose green tea in your example and not black tea. And just finally, I drink black tea in the morning and green tea in the afternoon.
Aha, so you like the caffeine, huh? And that's fine. Okay, so there are a lot of questions in there. First is, what's the difference between green tea and black tea? Black tea is fermented. So you take the tea leaves and you go through a fermentation process, and that fermentation process actually can polymerize the polyphenolic antioxidants. So they get bigger, they become tannins and things like this. And that's where the brown color comes from. And so there are two aspects here of antioxidant activity. The first one is, how much of the antioxidant is there in a food. And the second one is, how available is that when you digest it? How much of it do you absorb? And then the green tea, with the smaller antioxidants, because they haven't linked together in these kind of chain-like structures, those are better absorbed than the antioxidants from black tea, generally. Now, does that mean that black tea doesn't have any useful antioxidants? No, no way. I'm just saying that the smaller ones tend to be better absorbed. And there have been a whole lot of hype about you should drink this kind of tea or that kind of tea or eat this fruit. You know, it's kind of the fruit of the month club. Eat your blueberries, eat your acai berries, eat your, I'm not sure what kind of berries. And the answer is dietarily, what people know is if you eat a diet rich in fruits and vegetables and all these foods that I showed you on that chart and, you know, that have these antioxidants, chances are, statistically, you're at less risk for cancer and cardiovascular disease and all that. Does that mean you don't have to exercise? No. Does that mean, you know, you're never going to get it? No. But it means that generally people are healthier who eat more of these antioxidants. And as far as which antioxidants and in what foods, that's where we haven't been able to figure out if there's a correlation. You know, people who eat this, I don't know, live to be 100. There's no very clear correlation like that. So drink your green tea, drink your black tea, whatever floats your boat, just get your antioxidants. And there's an interesting corollary to the black tea question, because there were 2 papers actually published on the antioxidant properties of black tea. And one of them showed that if you put milk in black tea, that the antioxidants in black tea weren't absorbed as well. And so people were like, oh my goodness, you know, especially Britain, this caused a huge uproar in Britain because they like to put milk in their tea, right? And to the point where the British Cardiovascular Association had to issue a press release saying putting milk in your tea is not harmful to your health, right? And what the researchers had done, if you look carefully at the paper, is they had taken the black tea and put the milk in it and then centrifuged it down. And the proteins in the milk were bringing the polyphenols out of the tea. So I guess the moral of that story is don't centrifuge your tea before you drink it. But I don't know very many people who do that. So you've got to be really careful about these studies. There's a lot of weird things that go on, you know. So that's my long answer to, you know, which ones should you eat or drink or whatever. We don't know yet.
Thank you.
Mm-hmm.
I have a question. That was an awesome talk. My
Thanks.
undergrad was microbiology, so I love all of this stuff. But I have a question about the origin of iron. So can plant-based irons, like coming from spinach, are they somehow less or more or the same, like having the same oxidative stress as irons that are like heme irons, like from animal proteins?
Yeah, and there you have a little bit of the availability question. It's known that if you have a heme, like in hemoglobin, you know, red meat, these kinds of things, that iron is taken up better than some other forms of iron. You know, iron supplements in some maybe not top of the line cereals, you know, when they say iron supplementation, if you take a magnet and you hold it up to the cereal, you can actually get little iron filings up on the magnet because they've just sprinkled in iron filings into the cereal. That kind of iron supplement is not as bioavailable as something like, you know, heme and hemoglobin or something like this. And so you have this question of what exactly is the form of the iron that's taken up. And so, yeah, some foods have iron in a way that we can use it better than others, or, you know, iron filings.
So I guess that's like a trade-off then of like, you know, you need iron for some things, but you also don't want the oxidative stress that.
Right. Anemia is also a problem. You know, over the course of human history, anemia has been a bigger problem than iron, you know, getting too much iron. And if you have anemia, you don't have enough iron to adequately transport oxygen in your blood. And that's bad too. So it's always a balance. It's always a balance in biological systems. You don't want it too little, you don't want too much.
I just have one more question too, about like topical versus, you know, like ingesting your like phenolic acid. So I know that there are a lot of skincare products that have a lot of phenolic acid and that kind of thing in it, like green tea extract and all that kind of stuff.
And copper
Does that
too.
help with like antioxidant on the skin? Like, does it matter if it's topical, or should you be more concerned about getting your phenolic acids in your diet?
Personally, although I have not seen any particular research comparing the two, your skin is dead, right? I mean, the outer layer of your skin is dead. So if you're going to smear stuff on the skin, it's got to go through this dead layer that's supposed to protect. Whereas if you're going to eat it, it's going to go into your stomach, which is supposed to digest. And so if my goal were to get antioxidants, I would assume that it would be much more, they would be much more available if I ate them or drank them than if I put them on my skin. Now, you know, that could vary. I mean, if you're trying to, you know, anti-age your skin and you're putting it on your skin, some of it might get through, right? That outer layer of skin, and it might get into the dermis, you know, underneath. They do this with copper too. They put copper in a lot of these higher end anti-aging serums. And it turns out, because copper is one of the metals in collagen that helps collagen production. And so the idea is when you age, your collagen isn't as, it doesn't, you don't have as much collagen. And so you need more synthesis of collagen, hopefully. And so somehow putting copper on your dead skin will help that. You know, they say these things, they don't have to prove that this works. And so there just haven't been very many actual scientific type studies to show benefits of this.
So iron and copper are readily chelated by a variety of compounds. What role does that play?
Well, I mean, it plays a big role in our research because we have to chelate just about everything before we do our studies. And that's a pain in the rear end, because even like if you get a piece of dust in your solution, you have to throw it away because iron is a major component of dust. And so that's a pain. In some circles, chelation therapy is talked about, where you take these molecules that are supposed to bind up and you know, the metals, toxic, they call them toxic metals, and then you excrete them. And that's a bad idea. I mean, unless there's some specific reason why you know you have too much metal in your body of some type, do not do this to yourself, because one of the side effects of metal chelation is it can stop your heart, because your heart relies on calcium to beat, right? All your muscles rely on calcium to contract. It just so happens your heart is one of the most important, and if it stops beating, you're in trouble. So people who have metal toxicity issues, say you've been exposed to a large amount of metal ion, you go to the hospital, you say, I've had this metal exposure, and they will treat you with chelators, but they'll be very careful about it to make sure that they don't give you enough that will overload your system and start chelating the calcium out of your muscles. These are some really great questions.
Yeah, Julia, I noticed in the chat box, we do have a couple questions. I think a lot of them you kind of answered in your other explanation, but someone just asked about the impacts of water and the amount of water helping with free radicals in general.
Yeah, hang on, let me look at the question. Water doesn't help with the radical generation process, or it doesn't hinder the radical generation process particularly either. But I mean, it helps with hydration. because if you don't get enough water, you have a lot of other biological side effects. You know, I mean, your body is basically water encased in membranes and skin and stuff. And so you really need that for enough water for the biological processes. But it doesn't typically have an effect on radical production or anything like that. Let's see if there are any more. Oh, does sugar affect the amount of antioxidants in tea? No, the tea has the amount of antioxidants that come out of the tea bag, or you know, however you're making your tea. If you want to add sugar, that's not going to affect the amount of antioxidants or the absorption of antioxidants or anything. I personally think green tea tastes like grass, so I don't drink it plain. So it might make you drink more green tea if you think it tastes like grass. And so somebody asked, eating a large variety of vegetables and fruits is our best insurance. Yeah, that whole balanced diet with fruits and vegetables, that is the best advice that you know, people can give. And I'm sorry if that's not you know, what everybody was hoping for. A lot of people think they're looking for the magic bullet, and there's really not. Okay, these drinks with antioxidants like I've never heard of I have to go look that up. A marketing gimmick. Well, chances are those drinks have antioxidants. You know, it depends a little bit on how they're made and what they're made from and how much, because you always have the problem where, you know, it says that this is, I don't know, acai berry juice or something like this. And you look at the ingredients, it's mostly like apple juice and with a little bit of acai. So that's going to affect things. So you have to be careful what you're actually drinking, because they do get creative with their advertising. Let's put it this way. I once saw a bottle of 7-Up in the supermarket that advertised 7-Up now with antioxidants. And what they meant was the ascorbic acid they added for freshness was also vitamin C, which is an antioxidant. But if you're getting your antioxidants from 7-Up, you're probably on the wrong track dietarily. So they get creative with this. And so is it a marketing gimmick? I don't know about this particular one, but I would be at least a little skeptical and look into it a little more. Oh, bioavailability of polyphenols in vegetables versus a supplement. Yeah, that is bioavailability of polyphenols is an active area of research. People are looking at which polyphenols are bioavailable and where they're absorbed, like in the stomach versus the intestine, and how long they last in your bloodstream, and all of these kind of questions. And you can get green tea supplements, like pills, like, you know, concentrated green tea powder kind of things. They do have a lot of antioxidants in those pills. So I guess you could take those, and I know there have been actually a couple of incidences where people have done themselves harm by taking so many of those pills that they have, you know, caused medical problems. So yeah, I would vote for the fruits and vegetables way rather than, you know, trying to shortcut the situation with pills, because it's not a situation where more is better. More can be harmful. That's always true in any biological system.